Fats are pretty important. They're needed to make phospholipids, which make up cell membranes, lipoproteins in the liver, and even breast milk. But the primary role of fats is to serve as the main storage of energy. Fatty acid synthesis is the biochemical pathway of making fatty acids from excess carbohydrates. In this sketch, we'll go back to Sketchyland Pizza to understand how fatty acids are made. And check this out, they gave us all VIP access passes to the kitchen, so we're going to learn how these zigzag creations are made. But before we put on our pizza hats and aprons, let's review what fatty acids look like. Fatty acids are composed of a carboxyl group and a chain of hydrocarbons. As you can see from this diagram, they're classified based on the number of carbon atoms. Unless you live under a rock, you're probably familiar with saturated and unsaturated fat. As a reminder, saturated fats don't have any double bonds connecting the carbons, while unsaturated fats do. Some fatty acids are essential, meaning that our bodies can't make them and they need to be obtained from the diet. The essential fatty acids are the omega-3 fats, ALA, EPA, and DHA, and the omega-6 fat, linoleic acid. Most fatty acids are stored as triglycerides in adipose tissue. Recall that a triglyceride is made up of 3 fatty acids connected to a glycerol backbone. Alright, now that we've got all that general information out of the way, we can talk about how fatty acids are made. Fatty acids are made in the cytosol of adipose tissue, hepatocytes, and lactating mammary glands. After you eat a high-calorie, high-carb meal when there's excess glucose and lots of ATP inside cells, it's time to make fatty acids. Blood glucose levels rise, stimulating the release of insulin from the pancreatic beta cells. And insulin is the main driving force for fat synthesis. It moves glucose into cells where it can undergo glycolysis to make pyruvate before entering the mitochondrial matrix and becoming acetyl-CoA. What happens to acetyl-CoA? Well, it enters the TCA cycle and goes round and round and round until we make a bunch of ATP from oxidative phosphorylation. Eventually, ATP will slow down the TCA cycle and acetyl-CoA will start accumulating inside the mitochondria. What do we do with all that excess acetyl-CoA? Decisions. Well, while you're sitting there regretting your dietary decisions, your body's all, yo, I'm gonna turn that acetyl-CoA to fat and store it as energy just in case we enter a worldwide nuclear famine. And that, my friends, is why your body makes these decisions and not you. But that raises another question. If fatty acids are made in the cytosol and acetyl-CoA is hanging out in the mitochondria, then how do we transport acetyl-CoA out of the mitochondria? Acetyl-CoA can't just cross the membrane alone, but citrate, oh citrate, yes you can. Citrate synthase makes citrate from acetyl-CoA and oxaloacetate. Come grab a cup of OJ and watch citrate hop across the membrane like an Olympic hurdler. After citrate enters the cytosol, it gets converted back to acetyl-CoA. ATP citrate lyase uses ATP and CoA to cut up, or as you medical people like to say it, lyse citrate, making acetyl-CoA and oxaloacetate. That worker is all hopped up on cola and using an ATP-powered blender to lyse orange juice into a smoothie. Doing a fine job, sir. Keep up the good work. The products, acetyl-CoA and oxaloacetate, are represented by the acetyl cola and Oaxacan chocolate powder. Before we discuss what happens next with acetyl-CoA, let's briefly mention what happens to oxaloacetate. Oxaloacetate has to get back into the mitochondria, but like acetyl-CoA, it can't directly cross membranes. Instead, malate dehydrogenase turns oxaloacetate and NADH to malate and NAD+. Then, malic enzyme catalyzes the oxidative decarboxylation of malate to make pyruvate before entering the mitochondria. Note the pirate hat for pyruvate. Just a side note, this reaction also turns NAD+ to NADPH, or our recurring symbol of energy petrol. If you watched our pole position speedway sketch, you'll remember that NADPH is made during the pentose phosphate pathway. Spoiler alert, we need NADPH to make fatty acids. Okay, back to the real story. Let's continue on our journey with acetyl-CoA, which we just made by lysing citrate. The next step is very important. Acetyl-CoA carboxylase irreversibly uses ATP to add a CO2 group to acetyl-CoA, making malonyl-CoA. Do you want to know why the colas are extra bubbly here at Sketchyland Pizza? Look no further than the acetyl-cola carbonator. Like all carboxylase reactions, acetyl-CoA carboxylase requires vitamin B7, aka biotin. The product, malonyl-CoA, will be the substrate used to elongate fatty acids. Just a reminder, malonyl-CoA inhibits CAT1, the enzyme that turns fatty acyl-CoA into fatty acyl-carnitine, so it can enter the mitochondria and we can proceed with beta-oxidation of fatty acids. Let's talk about how acetyl-CoA carboxylase is regulated. Insulin activates acetyl-CoA carboxylase. Place your cup inside to get the carbonated beverage of your choice. Remember that inside is a recurring symbol for insulin. Citrate also activates acetyl-CoA carboxylase. High citrate levels means there's plenty of energy available inside cells, so we can make fats. On the other hand, glucagon, epinephrine, and palmitoyl-CoA inhibit acetyl-CoA carboxylase. Let the spilled glucagon sweetener, Epic Cup, and palm oil remind you of that. Finally, we can make zigzag pizzas. Now, you're probably wondering what the secret to make them is. Or probably not. Yeah, it's nothing special, just zigzag-shaped dough, sauce, cheese, salami, a little bit of love, and boom, taste of heaven. Making fats? Not so easy. It requires a large multi-enzyme complex called fatty acid synthase, or FAS. This complex is huge. FAS uses acetyl-CoA and many units of malonyl-CoA to make 16-carbon palmitic acid. While you don't need to memorize each specific enzyme, you should understand how FAS works. First is initiation. FAS contains two important substrate binding regions: a pantothenic group associated with an acyl carrier protein and a sulfhydryl group on cysteine. Acetyl-CoA first binds to the pantothenic group on the acyl carrier protein. Afterward, acetyl-CoA is transferred to the sulfhydryl group on cysteine, while malonyl-CoA takes acetyl-CoA's spot. Once both substrates are loaded, FAS can now catalyze a series of 4 reactions: condensation, reduction, dehydration, and reduction again. Each cycle adds 2 carbons to the growing fatty acyl-CoA and requires 2 NADPH. We repeat each cycle until we finally make 16-carbon palmitic acid. Don't like your pizza plain? Well, you can always add different toppings here at the topping station. It's got a bunch of clear tubes filled with different toppings. These tubes look just like the smoothie are, but not as hygienic. What a coincidence, because palmitic acid can undergo different modifications in the smoothie are. Palmitic acid first needs to be converted to palmitoyl-CoA before desaturases can add double bonds. This process is complex and you don't need to know what exactly happens, and I'm getting hangry right now, so we'll just finish up here after a quick outro. Fatty acids primarily serve as the main storage of energy. Fatty acid synthesis takes place in the cytosol of adipose tissue, liver, and lactating mammary glands. Fatty acids are made from excess carbohydrates when there's lots of ATP available inside cells. To make fatty acids, acetyl-CoA must first combine with oxaloacetate to make citrate. Next, citrate crosses the mitochondrial membrane to enter the cytosol before getting converted back to acetyl-CoA and oxaloacetate by ATP citrate lyase. Oxaloacetate undergoes 2 steps to make pyruvate before re-entering the mitochondria. Meanwhile, acetyl-CoA carboxylase irreversibly uses ATP to add a CO2 group to acetyl-CoA, making malonyl-CoA. Insulin and citrate activate, while glucagon, epinephrine, and palmitoyl-CoA inhibit acetyl-CoA carboxylase. Finally, fatty acid synthase makes 16-carbon palmitic acid from acetyl-CoA and malonyl-CoA.